The Maxam-Gilbert method is based on a chemical sequencing. The first step is the preparation of the single-stranded DNA. For this, we need to remove the 5' phosphate group with an alkaline phosphatase. Once we got a homogeneous strand, we add a radio-labeled phosphate group into the 5' end, using a polynucleotide kinase. After we add the phosphate group, we are ready to begin the chemical termination. This process is based on the addition of certain chemicals which will degrade and cleave certain bases of the DNA. It divides in 4 major reactions. In the guanine reaction, the addition of dimethyl sulfate followed by piperidine cleaves the guanine base out of the DNA strand, as shown in the reaction. In the adenine and guanine reaction, the addition of formic acid followed by piperidine will always cleave adenine, and sometimes guanine, as shown in the reaction. In the thymine and cytosine reaction, the addition of hydrazine followed by piperidine will always cleave thymine, and sometimes cytosine, as shown in the reaction. In the cytosine reaction, the addition of hydrazine and sodium chloride followed by piperidine cleaves the cytosine base out of the DNA strand. Different sized DNA strands in 4 different test tubes. Then, the fragments are electrophoresed in high-resolution polyacrylamide gels for size separation. These gels are...
First of all, you need to fragment the DNA. After that, oligonucleotide adapters are linked to each end of DNA. These adapter sequences common to all fragments will be used both to link each fragment to the spheres and as sequences where the PCR primers will join, but also presents the sequence where the primer of the sequence will be linked. Once the adapters are attached, they are linked to the bits that are spheres containing the complementary to one of the adapters that are on their surface. This is made by a limit dilution method to obtain in this way a single fragment attached to a sphere. It is sought then to obtain in each bit a single DNA fragment that is amplified by emulsion PCR. The methylation interference assay refers to the interaction between DNA and protein. It's a method of identifying which regions of DNA strands bind to specific proteins by methylation modification of nucleotides, so it can be used to study the relationship between transcription factors and guanine residues in DNA binding sites. Is to know the expression of genes according to external factors that activate them. This is made by extraction of RNA, synthesis, and labeling of complementary DNA, then hybridization, lecture and quantification, and the final analysis.
The Sanger method uses DNA polymerase and inhibitors that ends the DNA strain that is being synthesized in particular places. It uses the nucleotides that doesn't have in its carbon 3 the hydroxyl group.
How does Illumina sequencing works? First, the input sample must be cleaved into short sections. About 100 and 150 BP reads are used. The longer fragments are ligated to a generic adapter. Then they are annealed using the adapters, and the amplification of the lecture is done by PCR. Next, they are separated into single strands. In the image, we can see a graphic representation of how Illumina sequencing is done. The nucleotides are fluorescently labeled with the color corresponding to the base. For example, thymine with green, cytosine with red, adenine with yellow, and guanine with blue. Also, it must have a terminator, so that only one base is added at a time. Therefore, in every single read location, there will be a fluorescent signal indicating that the base has been added. After that, the terminator are removed. This allows to be added the next base, and the fluorescent signal is also removed. This is because preventing the contaminating of the next image. The process is repeated, adding one nucleotide at a time. The 4-5 sequencing can do longer reads than Illumina, but like in Illumina, it does sequencing by multiple reads at once, using optical signals. The short fragments are up to 1 kb. It also uses adapters that are added to the ends. These generic adapters are enabled to bit one DNA fragment per bit. Then the amplification occurs by PCR using adapter-specific primers, like is represented in the image. Therefore, each bit has a single well of a slide, and every single well covered in many PCR copies of a single sequence. It is important to mention that the slide is fluid with one of the four NTP species. When a nucleotide is added, it releases a light signal. This helps to determine which bits the nucleotides are added. Then the NTP mix is washed, and then they put again another NTP mix, and the process is repeated. This is through every single NTPs. A difference between 454 sequencing and Illumina sequencing is that ion torrent do not use optical signals. It uses the fact that the addition of a dNTP to a DNA polymer releases an hydrogen-positive ion. The DNA is fragmented in 200 PV, and also the amplification is done by the PCR. The slide is fluid with a single species of NTP along the buffers and the polymerase. When a hydrogen is released, the pH is detected. Every single hydrogen ion released will decrease the pH. This allows to determine if that base was added to the sequence read. The NTPs are washed, and the process is repeated with every single NTP species. The change in the pH allows to determine how many bases were added in every single cycle.
Relevant laboratory equipment such as the Pocket Minion is a powerful and portable sequencing device that can deliver large volumes of long-range sequencing data. The Gridion H5 workbench can group up to 5 million flow cells at a time, on demand, for larger genomic projects. The recently launched Prometheus is the largest format for nanopore sequencing, designed to offer on-demand use up to 48 flow cells, each of which can offer more than 100 GB of sequencing data. Minion marketing that was 2015, Gradient was in 2017, and Promotion is in 2018. However, the company is looking for a development that allows its use to any user, anywhere from mobile phones with low-cost portable samples with SMIC ion and Flonk.